Hi everyone, my name is Daniel Nakamura, a PhD student from the University of Sao Paulo. Today, I would like to talk about the impact of phenomic evidence on total evidence analysis dominated by DNA sequences. Phenomic characters, such as those related with external morphology, osteology, and bioacoustics, used to be the primary source of evidence in phylogenetic analysis in the first decades of logistics. However, with the advent of PCR and central sequencing, we now have the dominance of molecular sequences as the primary source of evidence for extant taxa. This trend has been reinforced since the invention of high-throughput DNA sequencing in the 2000s due to the declining costs and increasing speed for generation of large molecular datasets, as opposed to the small, time-intensive, taxon-specialized process of collecting a few morphological datasets. Although we have now ancient DNA from museum specimens, the use of phenomic evidence is important if you want to include fossils or specimens with no endogenous DNA. Despite this huge advantage of phenomics, their low number, typically from dozens to hundreds, relative to molecular characters, typically from thousands to millions, has sparked diverging opinions about the value of phenomics. On one hand, several authors have challenged the relevance of morphology to impact topologies and only analyze morphology based on molecular trees a posteriori using phylogenetic comparative methods. In contrast, many authors advocate in favor of a total evidence approach, in which morphology participates of phylogenetic inference. And the reason is simple for the total evidence approach. You don't need to ignore or even delete morphological data because the more empirical evidence you have, the more severe is your hypothesis test. Clade support is typically assessed using two types of metrics, those proportional to optimality, like Bremer, and those that rely on resampling, like Bootstrap and Jackknife. Resampling metrics are more popular in phylogenetics. Specifically, high values of Bootstrap are often interpreted as indicators of either high accuracy, the probability of a clade being true, or high repeatability, the probability of a clade being repeatedly recovered given new samples or given new data in the future or perturbation. But we know very little about the relationship between Bootstrap and the addition of phenomic data, especially if we consider the emergence of support when we have more data, the hidden support. For instance, we don't know if the incorporation of phenomic evidence have no effect increase or reduce Bootstrap. We also don't know if we can predict the occurrence of total evidence trees, or the occurrence of total evidence groups, the monophily of total evidence groups based on Bootstrap from molecular trees. So here in this study, using 57 empirical datasets comprising 90 to 500 leaves, 13 to 4,000 phenomic characters, and 500 to 2 million molecular characters, we performed phylogenetic analysis using two optimality criteria: parsimony in TNT and maximum likelihood in IQ3. For parsimony, we analyzed the molecular and total evidence trees. For maximum likelihood, we analyzed molecular trees and also total evidence trees, either using the bias correction or not using the bias correction in the MK model. This bias correction is necessary according to some authors because only morphological characters with variation are usually collected whereas molecular characters can either have variation or not. We don't sample molecular characters and morphological characters in the same way. We discriminate morphology. We only sample variants, theoretically, but in practice some people also collect invariants, and that's why I'm using both approaches. In our analysis, we compared trees by the number of shared clades, the number of unique clades, some topological distances metrics like SPR moves, Robson folds, cluster information distance, and also we assessed the number of polytomies to understand the tree resolution. And we were interested on 5 questions, 3 of them related to topology, 2 of them related to support, and we repeated all analysis with and without fossils. To help the task of comparing support of corresponding nodes between 2 trees, we wrote an R package called RNode, which enables to identify shared and unique nodes and create data frames of support values of corresponding nodes, which is useful to facilitate statistical analysis, regression, and logistical regressions. So the first question is, are topologies impacted by the addition of phenomic evidence? Our expectation was that morphology is able to impact topologies even with large data sets, with phylogenomic data sets, and our results indicate that yes, molecular and total evidence trees were different in 87% of our topologies, with a tendency for molecular and total evidence incongruences to be different from 0. And this happens even in some phylogenomic data sets, and it happens both in analysis with and without fossils. The second question is, are molecular and total evidence incongruences associated with the proportion of phenomic characters? And we had the hypothesis that the more phenomic characters we have, the more the impact of phenomic evidence on total evidence trees. And our results using non-parametric experiment tests indicated that all topological metrics are correlated with the proportion of phenomic characters in our analysis, in our optimality criteria, with or without fossils. As a consequence of these results, one possible interpretation is that morphology will be useless in the future when whole genomes are available for all organisms. But this will not be the case because morphology is the only way to include fossils, and even if we are concerned about the possible character dependency between morphology and regions of the genome that will code morphology, we have now some methods to cope with character dependence, so it will not be a problem in the future. The third question is the addition of phenomic evidence equally impactful on likelihood and parsimony topologies? The hypothesis was that parsimony could be more impacted by the morphology because in parsimony we don't have the MK model, which is a very simple model, very unrealistic model according to some authors. So our results indicated that no, the addition of phenomic evidence is not equally impactful, and the incongruences between molecular and total evidence trees are higher in parsimony than likelihood, at least according to the cluster information distance. And this impact and this difference between parsimony and likelihood is greater when we consider the fossils. When we consider the fossils, the morphology is even more impactful in parsimony than likelihood. And one very interesting result is that the tree resolution is significantly different between molecular and total evidence in parsimony trees, but never in likelihood trees, which can explain the molecular total evidence incongruences. The fourth question is, how do bootstrap values of shared clades differ between molecular and total evidence trees? The addition of phenomic evidence tends to slightly increase bootstrap values in all analyses without fossils, but the bootstrap values are slightly lower in total evidence trees from parsimony with fossils. We didn't find difference in likelihood in the bootstrap values of likelihood between molecular and total evidence trees when we consider the fossils. And also, as expected, we always found the unique clades, they tend to be lower than the bootstrap of shared clades, which is expected. The fifth and the final question is, can bootstrap from molecular clades predict the presence of total evidence clades? And our results using logistic regressions indicate that the bootstrap values in molecular trees are positively associated with the occurrence of the same clades in total evidence trees. So we can predict there is a tendency, but there are some cases of molecular clades with 100% bootstrap values that are absent in the total evidence trees. So in these cases, the 100% value is no guarantee that the clade with 100% from molecular tree will be present in the total evidence tree. So the 100% is meaningless, according to some people, some authors. And that's one of the criticisms against bootstrap. Finally, the take-home message in this study, we compared molecular and total evidence trees in terms of topology and support. In topologies, phenomic evidence impacts topologies. The more phenomic evidence you have, the more impactful. But even in phylogenomic datasets, we still can detect the impact of morphology on total evidence trees. And the impact is higher in parsimony than maximum likelihood, according to some metrics like cluster information distance and Robinson folds. In support, phenomic tends to increase bootstrapping in parsimony likelihood without fossils, but weakly decrease bootstrap in parsimony with fossils. The higher the bootstrap in molecular clades, the higher the chances to recover that clade in total evidence trees. But very importantly, 100% values of bootstrap are no guarantee that you will find that clade again in the future or with morphology. Hidden support emerges when we incorporate morphology, which is another reason to collect morphological data and avoid a reductionist view that the molecular data alone suffice. So thank you very much.